Okay, good. Okay, and what we're going to do now is just watch all the participant numbers increase. We'll give it a minute to let everybody come on, and then we'll get started. Okay, I'm just kind of waiting for the numbers to stabilize, but they keep going, but I think we're going to get started now. So here we go. Okay, hello everyone, and welcome to the November ACS San Francisco Bay monthly speaker event. And this is the last of 2021. I'm Susan Hopp, a board member, and I'm here with fellow board member Izzy Chapagnac. And for anyone new to the American Cetacean Society, I just want to give a quick introduction. And ACS began in 1967 and is the oldest nonprofit dedicated to the protection of whales, dolphins, porpoises, and their habitat. And we do this through education, community engagement, and grants toward marine research. And regarding our grants, we have some exciting announcements coming up, so stay tuned for that. And that brings us to our appreciation, for your donations in support of our mission. And they certainly help fund our research grants and support our expenses around these monthly talks. And if people just gave $5 or $10 or $20, it really sets us up to fulfill our mission and really do the work that we're all here for. And so all of you who do donate, thank you so much. So we're recording this session, and we encourage you to put your questions in the Q&A. So the Q&A is better than the chat. And after the presentation, we will get to as many of them as possible. And so we're really excited tonight. We're closing out our 2021 speaker program very fittingly with our talk tonight entitled All the Humpback Whales of the Pacific Ocean. Our speaker tonight is Ted Cheeseman, co-founder and director of Happywhale. And a little bit about Ted. So Ted founded Happywhale in 2015, but his story leading up to it starts when he was a child growing up and whale watching in California's Monterey Bay. In 1980, his parents, both naturalists and educators, founded Cheeseman's Ecology Safaris. In 1994, Ted joined the company as a leader of ecology-based wildlife safaris, working on all 7 continents. After earning a Master's of Science in Conservation Biology from Duke University, Ted returned to the company to lead polar expeditions. Since that time, Ted has been deeply involved in all aspects of Antarctic tourism management. He served on the executive committee of the International Association of Antarctic Tour Operators for 5 years, where he helped minimize the environmental impact of tourism in Antarctica. And the 3 decades in ecotourism gave Ted firsthand experience on the human impact and the challenges facing marine wildlife. In 2015, Ted stepped down from his role with Cheeseman's Ecology Safaris to work on marine conservation, and specifically capturing and using scientific data to further our understanding and protection of whales. Happywhale was born out of Ted's desire to create an innovative citizen science program that helps us better understand whales as individuals. So Ted, thanks so much for being here, and we're handing it off to you.
Well, cheers. Thank you. And yeah, no, it's really special. And thank you for, it's really special to talk to the American Cetacean Society, because I think ACS is especially ACS San Francisco, it's really in this nice space where there's a lot of science knowledge, a lot of scientists, but also the really enthusiastic and well-informed non-scientists or serious naturalists and amateur naturalists. And it's in this space, I think that it's, for me, honestly, it's a privilege, I feel, to be able to give something to this community, because I think it's here that science communication really happens in the most powerful way, as far as where, we have this ideal that good science can inform policy, and good policy can better protect the oceans. And it is sometimes a myth, but this is the kind of community I feel that can actually make it a reality. And when I go, Sunday, I was guiding out in Monterey Bay, and every time I'm out in the Monterey Bay Marine Sanctuary, it's like the visceral reminder, because when I was a kid, there were not humpback whales here. We did not go whale watching in the summer. The whales were not here, right? So we as a society have chosen to protect this environment. And it's honestly, I feel this is the community that has made and continues to make those choices. So, so it's a real treat to share. Thank you for that very flattering introduction. I'm going to share my screen and start my little talk here. And this talk, it's rather, well, I suppose it's a little bit preposterous to say all the humpback whales of the Pacific Ocean. But what we have, what I'm here to talk about really is the non-science conference version of the talk that I would have been giving next month at the Marine Mammal Society of Marine Mammals conference, had it not been moved from December in Florida to next August, I guess it is. And this is basically the second chapter of my PhD thesis. I'm about a year out from finishing my thesis, or maybe in a year I'll still be saying that I'm a year out, but in concept, I have a lot more to write, but I'm a long way through my thesis. The first chapter of this here thesis was the building of automated image recognition. So the tails that you see. So by way of introduction, what I'd like to do is kind of give you the brief synopsis of what we've done in terms of technological development, and then really focus on kind of this gathering together of an ocean basins full of whales and all the collaborators that made that possible. So by way of that introduction, I mean, first things first, I think there's probably not many people on this call who aren't already aware, or on this talk that aren't already aware of this, but humpback whales, well, pretty much every animal is in some manner individually identifiable. Well, actually, every animal is in some way individually identifiable. And identification of individuals is an extremely powerful tool for science. There's lots of different ways we can use that. There's lots of different ways we can access it. Every animal is individually recognizable, certainly by their genome. That is rather difficult to access. Humpback whales, in this photo tries to illustrate, all have individually recognizable, the underside of their tails, the ventral surface of their tails have different patterns and shapes, every single one unique, like a fingerprint or a face. And in very brief, what we've done is automated that so that a technique that's now 50 some years old, manually matching individual flukes one against each other, well, that technique became extremely challenging at a certain scale, where once you have thousands of individuals, it might take an hour to determine, yes, this animal is new, and 10 or 20 minutes to find a match if the animal is known. Okay, we haven't found a match, then you have to keep searching until you're confident this animal is new. Well, we've managed to automate this into using artificial intelligence, machine learning, deep convolutional neural networks to take that hour and do it in literally a tenth of a second. And so with that, been able to build a increasingly global catalog. And a big part of this, and normally this kind of slide, who are the collaborators? Who are the contributors? Normally it's at the end of the talk, and I've usually left it there, but I think actually for the nature of this content, it's really important to bring this up to the front, which is that what I'm doing is really, it's this proverbial standing on the shoulders of giants, right? I mean, I'm kind of a Johnny come lately in terms of the marine mammal science, and I've been able to do what I've been able to do. And it's I use the word I, but I really should be saying we. In most particular, my partner Ken, the programmer behind all of this, and a whole host of other folks, the data managers behind Happywhale, and then this long list of collaborators, too long to even mention those here. And then there's at the bottom here, you can see, and 8,000 citizen scientists. The papers that we're writing out of this, I'm sort of insisting, including a appendix section that has literally pages and pages and pages of people listed who have contributed one or more photos to this. But basically the core and the engine of what we've done, again, about the image recognition is matching individuals over time and space. There's lots of different ways to follow a whale. You can do it with a digital tag and some fantastic science. Many of you probably will be familiar with this paper that's recently come out from, well, gosh, I think there were 17 co-authors, but Ari Friedlander, who's on my graduate committee, was one of the co-authors, where they found, hey, look, these whales are eating a lot more than we thought they were. Well, one of the key techniques there was using digital tags, suction cup tags on these whales to follow their movements in high resolution in time and space over a day, 2 days, 3 days, right? That's at one extreme. Somewhere in the middle are various kinds of telemetry that stay on the whale for a while, but even the most solidly implanted satellite tag will only stay on the whale for a few months. What we have with photo ID is, well, wonderfully a non-invasive technique, but also something that lasts over the course of the entire lifetime of an individual. And in this case, I'm showing two difficult to match cases. The top one is a whale that has two nicknames because it was seen here in Monterey Bay as a juvenile, as a first year, not a juvenile, as a first year calf with its mom, was named Pepper because it had this horrible skin condition. And I'd be out on the whale watch trip and we'd see Pepper and isn't this wonderful? Yay. And we call this whale Pepper. And then I'd turn the microphone off and say, that whale's going to die. It's in the worst body condition. And sure enough, though, showed up the next year, given a different name, the whale on the top right there, named Vertigo. And then this was a few years ago. And then with the advancement of our algorithm, we reanalyzed, we've consistently reanalyzed the data set and found, hey, wait, these are the same whales, because the change over time, first summer individual and to be able to see calves on up through adults and tracking them individual, that's a real treat. The case below was actually a whale with two different IDs that sat in a catalog, manually matched for 30 years. Every time a whale was taken through this catalog, they looked at both of those images and didn't realize, in fact, these are the same whales, just because the nature of manual matching. Well, we miss things too with the computer vision, but it is proven to be a fair bit more accurate. And that robustness has been pretty powerful, and I'm really excited about it, because what it's allowed us to do is not just do things faster, but also push into some margins where we weren't able to match so well before. And those are actually, so what we found with this algorithm is we did an accuracy assessment. We find that we can detect about 97 to 99% of potential matches. Now, most studies that have done accuracy assessments have benchmarks of like 6 to 10% of potential matches missed. So good bit better there. And that difference, well, the difference in those matches that you miss, they're not just like any whales. It's kind of non-random, right? You're not going to miss the obvious pattern ones. You're going to miss the ones with minimal patterns, poorly demarcated flukes or highly changed flukes or poorly presented flukes. And it's that last case that to me is really important. Top left here, you've got an entangled whale. This whale was actually cut free, but kind of a pretty poor quality fluke, partially submerged, and so on and so forth. Well, we matched this whale to the individual on the top right, and not only was it a poor quality fluke, but it was also a highly changed fluke, it turns out, on the top right there. That was the whale as a first summer individual. So, basically a calf. So got to, in fact, follow this whale and be confident that hopefully, I'm not sure off the top of my head if this whale has continued to be sighted on through the years down the road. But to be able to track the survivorship of whales seen entangled is, given that the Monterey area crab season is closed right now for the protection of whales. Well, the reason for that is expectation that it is harmful for whales, and trying to assess that and understand what is the best route to navigate management of a fishery that can be deadly to the whales, but also is an important fishery. I find this pretty valuable and important. So just before we got on the public, opened up this talk, Izzy here from ACS said, "Oh, the splash map, boy, that map gets a lot of use." So this study, SPLASH, the structures of population, levels of abundance and status of humpbacks of the North Pacific, is basically the landmark study that we are building on with this work. And I found this study hugely inspiring. It took 18,000 different encounters, manually matched them, found 8,000 whales in the set, and I had the sudden realization that I might be sharing the wrong screen. Are you seeing just the splash map, or are you seeing my next slide too? Anyone?
Just seeing the splash map right
Good.
now.
Okay. I just sorry. Anyway, cool. Yeah. So 18,000 encounters, 8,000 whales, and before the splash study, I mean, it's amazing to me because humpbacks are like the best studied large baleen whale, right? And yet the depth of our ignorance is really profound. Prior to this study, the general line of thinking was that California humpbacks, they might feed a bit here, but they were basically on their way up to Alaska, right? And then we realized with the study, hey, we have, oh, my mouse doesn't show up here. That's too bad. We have a distinct population along the California coast. It is, these whales have site fidelity to feeding areas. You go up to British Columbia, southern British Columbia, northern Washington, mostly different whales from along the California coast, up into Alaska, southeast Alaska, different individuals, and so on and so forth. But Splash is now 15 years, 16 years old. And that population, the North Pacific population, has been growing pretty rapidly in this time and growing at different rates across the Pacific. So the idea of my work was really for one, to build upon the Splash study, but also a big part of it, and I have not yet been criticized in my thesis work for being not ambitious enough. I, for some reason, decided that the North Pacific was not enough. Well, the reason, actually a very strong reason, which is that I've been working in Antarctica now for I'm going to be heading south in a couple of weeks. And I think it'll be my 37th year. Am I doing my math right? Yeah, pretty much. No, 27, sorry, 27 years. It's been a while, right? And when I first started going to Antarctica, the little island of South Georgia, which is in the top right of the map there, the glorious, most beautiful place on the planet, if you ask me. The first 16 years that we traveled to South Georgia, I did not see a single nearshore cetacean. Now, okay, so not a lot of whales in those waters, but what's really striking is the history is the 3 highest capacity whaling stations ever built were on South Georgia. And I think the next two largest whaling stations in South Georgia are among the top 10. The number of whales, there were, I think, 120,000 whales killed in the immediate vicinity of South Georgia. And for perspective, if you put the tip of South Georgia and the tip of at the southern limit of the Golden Gate Bridge, the south end of South Georgia would reach down what, halfway through Big Sur? So imagine if 120,000 whales were taken in that much of California's waters, right? We have about 3,000 humpback whales here. We have some thousands of gray whales that pass through our waters. We have about maybe 2,000 blue whales along here. Like, not even close to that, not even an order of magnitude of that number exists in our very, very rich marine ecosystem here. So, but in those early years, in the 90s, we did see some humpback whales along the Antarctic Peninsula. It was a really special event when we did. We would have some whale watching event per each trip, but it wasn't, you know, all the time. Whereas now, along the Antarctic Peninsula and the areas that we roam along, or that kind of that top third of the Antarctic Peninsula you see there, well, now it's just chock-a-block with whales, which is just so lovely. But this kind of map, I mean, this blank map is essentially the state of my knowledge of whale populations when I first started going south there. And quite frankly, in terms of global knowledge and scientific knowledge, well, yes, there is a lot of good science there, but boy, it's spotty. So my whole perspective was, can we take the really refined understanding of population structure and abundance in the North Pacific? Can we update it, because we know we have a lot of resources, but also can we use that and piece together the much slimmer data sets that are available in the South and come up with some element of that. I kind of got a little ahead of myself in talking about the whaling in the Antarctic. This is Grytviken Whaling Station, the first. I'm sure, you know, there's probably a dozen people at least at here tonight who've been to Grytviken. Well, it was the first few years of whaling there in South Georgia was only inside the bays. Cumberland Bay is what you can see in the background of this. Nobody sees whales inside these bays now. Humpbacks are starting to rediscover these bays now, which is, I think, a really special time in kind of the history of human and nature interface here. And I'm not going to dwell on graphs too much, but this thing is just too extreme to pass by. You know, the left side of the map, you've got basically the pre-whale, or left side of this graph, the pre-whaling population that we think it was. And this is from Zerbini et al., Alex Zerbini from NOAA up north, up in Seattle. Fantastic scientists, did some really great work with a bunch of collaborators and put together historical estimates of the better part of 30,000 individuals in the South Atlantic. Then the whaling period and industrial whaling for great whales really was a mining event, right? It was, we came, we clear cut, we mined, we extract, we you know, it was sort of open pit mining, as it were, just took all the whales out and then they were gone. They were commercially extinct. Most of the products made from whales were like margarine, dog food, fertilizer. We're not talking about high value products. And fortunately for the whales, for most species of great whales, the right whales, North Pacific, North Atlantic right whales accepted, but for most of the whales, they were commercially extinct before they were biologically extinct. And I think what I really want to use this graph to speak to is we have this notion that these populations will come back to their pre-whaling numbers. But we need to realize that the oceans are different places than they were. These are urbanized, noisy, warmed, you know, pot trap riddled, ship traversed oceans that now that the whales are recovering into. And I look at our jobs as conservation scientists to try to reconcile these things. Is it possible for the whales to survive? And what do we have to do, hopefully, as much as we can have agency? What do we have to do? What can we do to make the oceans healthy for the whales. And in fact, as we're increasingly coming to understand, the whales are part of making these oceans healthy. We did quite a number on the whales, and I won't belabor that too much because I think I'm talking longer than I need to already. But unfortunately, it took a really, really long time to come to any international ban. It was really only because they were virtually commercially extinct and their products were becoming cheaper at the same time. So there was becoming, not a profitable endeavor that the nations of the world signed international whaling moratorium. Definitely, unfortunately, especially for the right whales. Russia, everyone signed, as well as Russia signed these conventions, and Russia kept on whaling. I don't say this to point a finger at Russia, but just to kind of point out some of these little blips later on along that red line graph at the bottom, where some of the Russian whaling, Americans were very much pioneers of whaling technology until we discovered oil, and then our focus went elsewhere. Handed on the torch to Norway, and on down the road. And the last torchbearers were certainly Japan, Iceland, Norway, and Russia. And the Russian illegal whaling was a bit of a strip mining event, and something that, as a collective society, this is pretty much what we have, is like, find the hidden history lesson, right? We had, back in the day, bountiful catches and a relatively skinny human hunger, and now we have these rather slender catches but a rather more engorged appetite as a society. So a lot of what I've been trying to do is really take this rather whiz-bangy, fancy magic black box that we've developed that matches all the whales, and push it into the conservation science, because like I say, we have a very different ocean that the whales are trying to recover into. And I'm sorry for the gory image, but it does seem to be, something that gets people to pay attention. There atop this North Atlantic right whale is Michael Moore, one of my true heroes. Right, if you're interested in moderately depressing but definitely important reading, he has a new book out, Michael does, called oh gosh, what is it? We Are All Whalers, or something to that effect. It's basically about the modern environment that these whales are recovering or not recovering into. Ship strikes, entanglements, these are the two most direct and impactful causes of mortality of whales today. It's really easy to get up in arms about whaling. I am definitely no fan of commercial whaling, but it's a well, it's a bit of a pun maybe, but it's a total red herring issue when you take ship strikes and entanglements by the numbers and by the indiscriminate impact, certainly by the animal cruelty issues of this. This is a whale by the name of Scarlet, who managed to throw the entanglement that she got caught up in, a crab pot that she carried for a few months. She managed to get herself free of it, but it basically had her entangled for most of the feeding season. And by the time that she was free, she was pretty wasted away. She was last seen, I believe, in November of 2016, and in California, disappeared, showed up again in April, completely emaciated, and then died shortly thereafter. These two things are, very easy to be out of sight, out of mind, but they're a really major factor in California. I mean, gosh, we have humpback whales feeding in large numbers right at the mouth of San Francisco Bay, and all of the lovely devices that we are watching this talk on came, many of them through that gate on a ship like this, and putting whales at risk to do so. So how do we make these spaces safe for the whales, and at the same time manage things in light of a very changing ocean temperature and pH regime? We whale watchers benefited from 14, 15, 16, 17, the whales were coming in so close to shore, you could like go horseback riding and whale watching in Moss Landing. And I think it's pretty clear now that for us, it didn't have a massive impact on, whale numbers directly, but it did dramatically increase entanglement events. And up north in the Gulf of Alaska, Southeast Alaska, we're now putting together the data. I mean, there's been some smaller pieces of it and some really important pieces of it, like Glacier Bay had a total calving collapse during the years of the blob, which was the largest marine heat wave ever recorded on the planet. But in terms of ocean basin wide, what I'm really excited to do is take basically this kind of knowledge, this kind of resolution, but extended over larger areas and a longer period of time. So this is an image of all the data in Happywhale. It's a bit old now, maybe a year old, I think, when I created this map. But specifically zooming in on the North Pacific, we now have so much data. We have an approximately, let's see, I think we have 28,000 individually identified humpbacks in the North Pacific. And those 28,000 are represented in close to 200,000 identified encounters. So what you have there is approximately 7 encounters per individual on average. Some of those whales have been seen 500 times. Many of those whales have only been seen once. But with that, we can start to tease apart pieces of this. And I want to loop back to that slide right at the top about collaborators. This has taken the work of so many people. I mean, it's funny to me, as a PhD student, part of my reason for doing a PhD was to be out among these animals and seeing them with sort of pressing questions. Like, I want to watch these whales and learn something. But the reality is that when I go out to sea, it's a distraction to the real work, because maybe I'll identify 5 or 6 whales, or 10 and 20 in a great day, right? Whereas sitting at home on the computer, I might identify and manage the data for 1,000 encounters or something like that. Obviously, time observing the whales, well, that's really important too. But especially here in the Eastern Pacific and Central Pacific, from Hawaii east and from kind of Gulf of Alaska east, we now have this really high resolution data set through which we're able to start picking apart to see, okay, what happened with the blob. And, we're still looking retrospectively, what happened in 2014. Where I'd really like to be is, and where I believe with the pieces we're putting together, we'll be able to say, okay, hey, look, this is an anomalously hot year we're entering right now. Fortunately, this year is not. But when the next one comes, not if, but when, can we see in much closer to real time what is actually happening, so that it's not a retrospective, oh, hey, if we did this management change during this time, we could have reduced the impact. What you have is a situation like with the North Atlantic right whale. Because of an anomalously warm year, the whales went to a different environment, right? Where they weren't so protected, ship strike rates went way up, entanglement rates went way up, and management was slow to get in on the game and change rules around it. And consequently, the species has declined by 20% in the last 4 years. We really, really want to avoid that. So, and then from that, extending onward to seeing these kind of larger environments that have been very, very data poor. And for me, this is a really fun thing to look at, because you'll recall that photo or that map earlier on, that composite satellite image where this was basically blank. And certainly there were many biologists who had big pieces of this, but a lot of this, and you'll notice the one blank between Australia and Africa, well, that's just the data poor area. It is quite clear now that whales in the Southern Hemisphere have basically circumpolar connectivity, whereas we still have not found a single whale that has actually moved from the North Pacific population to South Pacific or vice versa. And I understand the same to be the case with the North and South Atlantic. I might be wrong about that. Not so familiar with that literature. But so now we're at this far right end of the curve of, whales are now in various stages of humpbacks, that is, are in various stages of recovery. There are determined to be 14 distinct population segments around the world. That's something we could talk about for another hour or two. How legit is that? But of those 14 distinct populations, a few are still considered endangered. Our California whales almost entirely go, let's see, well, geez, all the lines on here make it pretty impossible to see. But our California whales, maybe 1 in 1,000 goes to Hawaii. Almost all of our whales are going to Mexico, and maybe a third or a quarter of them are carrying on to Central America. Now, that Hawaii population is deemed to be fully recovered, but if the blob had the impact that I think it did, that population really may have taken it pretty severely. At least the Hawaii migrating to Alaska, Gulf of Alaska, Southeast Alaska whales. I don't know. Well, I hope hopefully in a year from now I'm saying whatever I have to say with a good deal of certainty. But that population delisted entirely, believed to be fully recovered. The Mexican California humpback whale populations being believed to be still threatened, but recovering quite well. Whereas the Central American population is still listed as endangered, and we are not really sure. Are they not recovering? Is something else going on? Are they shifting on up to Central America? I have some pretty strong ideas about it, but there's a whole bunch of papers in preparation and in review and this and that and the other thing that I'm not going to jump ahead of by talking about it too far now. So, but I guess what's really, really important to me is kind of all the different pieces that this pulls together. And one of these is, with blue whales in California, we have good reason to believe that ship strikes are at high enough of a rate as to be stopping the whales from achieving their full recovery, right? Is that the case with humpbacks? Well, we're seeing pretty good population growth, but hey, this Central American population, if they're the ones that are endangered and they are, appearing to not, at least what we're seeing is that the numbers that are showing up in Central America, well, those whales preferentially use and feed in Southern and Central California. Well, it may not be a coincidence that those whales are traveling further through more urbanized waters and feeding in some of the most urbanized and heavily fished waters of anywhere that humpbacks live globally. So whether that's a tight connection or not, whether that's correlation or causation, I can't say yet, but piecing this all together, I guess I want to close with touching on a couple of my favorite studies that really I'm just kind of a somewhat minor co-author of, because it's really the great work of Collaborators, and I'm sort of facilitating, but it's the work of others. And that's like my friend Nico down in she's lives in Australia, Murdoch University, but studying in Mexico in Sayulita, Vallarta, Banderas Bay. Well, in Banderas, we've been able to identify different populations. You got whales that migrate all the way from Russia. And what this map shows is her data set, all the sightings of whales that she has cited. Not all of her sightings, well, it has those, but take all of her whales and look more broadly at all the sightings of the whales that she has photographed. So sorry, I said that really in a messy way, but you've got, a sizable number of Russian whales showing up in Banderas Bay. You've got whales from the Aleutians, from Gulf of Alaska, from Southeast Alaska, from British Columbia, from California. And are they just panmitic? Are they equally mixing there? Or are they doing different things and maintaining some level of separation? Well, one of the coolest things we have is, a paper that's in preparation is looking at she's actually found, and she and a few other folks, it's already been published about, that there's some whales that are feeding in Banderas Bay. In southern part of Banderas Bay has some quite deep water. There's some whales feeding there, which is totally, this feeding on the breeding grounds. They're feeding at lower latitudes. Kind of defies our whole concept of it now. But why are they feeding there, and how much are they feeding there? And one of the things that I find really fascinating is who is feeding there. And I can't spill her beans, but it's not a random set of whales that are feeding in Banderas Bay. It's a specific set of individuals, and it correlates to their breeding and migratory biology. It's really, really neat stuff. And then, so I'll close with what is my single favorite study that this is supporting. And I look forward to, whenever Martin Van Aswegen, the lead of this study, or Lars Beider talks about this stuff. University of Hawaii Manoa PhD student Martin Van Aswegen has fly a drone out over a whale, really beautiful imagery, and from that, taking a high resolution laser altimeter, you can do some basic geometry and trigonometry and come up with a body mass of a whale. And then from that, working with some data, some of which pulls from the whaling days, come up with a percent body fat and measure the condition of these whales. Well, that's really neat. And it's really neat in a moment. But what if you can do that over time? Well, if you follow the whale that you have photographed with a drone, you get a fluke photo, and then follow that whale over time. Well, Andy Szabo, who's one of the collaborators in this, Andy Szabo from Alaska Whale Foundation, he told me, gosh, it was yesterday, a couple of days ago, he said, we're actually using the data that we get from all the contributors, citizen scientists, et cetera, in Southeast Alaska to say, oh, here's that whale that we need a sample of. Now we can go and find him, right, or her. And to see a mother with a calf and watch the actual body mass of a mother be transferred to a calf and learn their growth rates and their wasting rates and their recovery rates on the feeding ground. I mean, this stuff just gives me goosebumps. So it's a real treat to have a part in enabling it with the high resolution automated image recognition and the collaborative gathering of data that we've brought together. So with that, I totally, it's all about the collaboration, which in and of itself has been certainly the most complex part of it. There's a lot of personalities in the whale science world, and I love it. It's fun, it's exciting, but and sometimes a little frustrating, but a huge tip of my hat to all who have stepped in and collaborated here. And with that, I'd love to answer any questions if I haven't talked beyond my window of opportunity here. So thank you.
Okay, thank you, Ted. This is Izzy. I do have some questions. One question is, how do you know matches are correct, especially the difficult ones?
Okay, let's see. How about a live demo? Isn't that the sort of thing you're never supposed to do in such an environment? Let's see here. I'll
This is a forgiving crowd.
Yeah, well, I hope so. Easy to here. Let's see if I can just share that one window. Here we go. Okay, so you're seeing this thing that says whale ID, right?
Yes.
Okay, I'm just gonna you asked me this and I'm not prepared, so let me find something to match against here. Okay, so this is I'm just going to take a random set of let's not take too many images. Okay, 28 images. A little set of whales from the Salish Sea in the last 2019, 2020. So boom, they're all uploaded. Okay, we'll identify them. The upload down and data transfer part takes a little bit of time, but roughly it's about 1/10 of a second per image. So boom, there's the results. We've got proposed matches of 27 of those 28 photos. One is proposed as with the system does not believe that it has found a match. And that is determined by way of this score, which is basically a proximity score. It's basically saying, like, imagine these photos thrown up into, or imagine, okay, each of these, I'm going to take an obvious match here. Okay, so here you go. Right. So take this photo, which is one of 5 in our reference set, right? And give this to a computer and say, okay, you have here a whale photo, describe it, right? So it's basically determining, it's doing a whole bunch of math to determine the edge shape and the feature shapes and blah, 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 blah. Plot that in a multi-dimensional space and just imagine it in sort of a 3D space, right? So there's this little cloud of features. And then we throw this image in it and we say, okay, do the same process to this image, describe that. I don't know if you can see when I say this, I'm saying, I mean the left image here. I don't know if my mouse is showing up. Then describe this image and throw it into that multi-dimensional space that has descriptors for all of the 64,000 whales that this was just matched against. And give me any whales that show up with a high score. The closer it is to that sort of cloud of features, the higher the score. And in this case, 0.91, like 2 quality images with a score that high, guaranteed it's a match, right? We still manually match. We still manually confirm every image. I do not go and search for whales, but I do definitely look at, so what I'll do here is I'll say, okay, let's ignore this threshold. And that threshold is a unitless number. It's 0.385. Shoot, I'm sorry, this isn't displaying quite correctly, but there's one image in these 28 that is proposed as to not match. I'm just kind of clicking through here and, having looked at thousands and thousands and tens of thousands of images, I've become very, very quick at saying, yeah, that's a match. Yes, that's a match. Yes, that's a match. And just kind of having a pretty good solid ability to spot when it is or is not a match. You can see these reference images, date back a long ways. So I'm just looking for that one that is potentially not a match. So here we go. Here is the one case that it was like, whoa, I don't know, it's below this threshold of 0.385. So it's saying, not sure if this is a match. And I do not believe that it is. I mean, the obvious thing is that barnacle scar it doesn't appear to be there in this photo, but it's mostly because of the poor presentation. I suspect that this is a whale that we know. It's just somewhat poorly presented. Does that answer your question?
Well, I don't know. I didn't ask that question, but
or how do we know that it is a match? Wasn't that it?
Yeah. So
actually, the Anastasia who asked the question just responded yes.
Okay, cool. Oh,
you
okay.
have
I thought
answered
that was your question, Izzy. Cool. Good.
Okay.
Good.
I see that. So another question, Ted. You just showed us how you do the matching there, and suppose one of the animals that's in your catalog and gets attacked by an orca and has bite marks on its flukes that were never there. How does your system take that into consideration when considering a match?
Yeah, good question. Change over time is one of the big challenges to photo ID for sure. Is you need a feature that is both persistent and accessible. And humpback whale tails absolutely change quite a bit in the first few years. Most killer whale attacks happen when they're quite young. And for those who don't know, humpbacks do tend to scar white on black and black on white. It's a really, very user-friendly feature of theirs. But this has proven to be pretty robust, and I think a big part of that is we can't really know how many we missed, but what I have come up with is like the splash study, for example, 8,000 individuals, and they estimated by taking a subset and looking really, really carefully, they estimated that they missed 9 to 10% of potential matches. Well, when we ran all the splash images through this system, we found about 8% of potential. We found about 8% of their data set was, multiple individuals that hadn't been, or sorry, single individuals with multiple IDs, right? So we found, I think, 8% of their estimated 9 to 10%. So it's pretty good. It's pretty robust. Here's an example of that. I think if you can see my screen on the right, you've got what's pretty certain to be a first-year animal. This was taken in 2015 on the Silver Bank. So probably a first-year individual. And then the photo on the left was taken this past summer up in somewhere in Canadian waters. I can't remember exactly where. So this isn't killer whale rake marks, but it's the same kind of change over time. We can't know what we missed, but it seems to be pretty resilient against that kind of change, and more so than the human eye, where we tend to focus on, we'll focus on this major pattern, right? The big blotchiness of, and the sort of the black and whites of the photo on the left, whereas the algorithm is going past that to see these smaller scars and the small features of the trailing edge. And so picking up on multiple features that we won't necessarily ourselves focus in on the human eye. Yeah.
Okay. This is basically, you showed a picture of the blue whale fluke. Yes. That John took. And, it was a pretty distinct fluke, yeah, which is unusual for a blue whale, but can this program be applied to other species of cetaceans like blue whales?
So good question. The specific, this touches on something I'm super excited about. Okay, the algorithm that we are using was built in collaboration with Google by a startup that Google bought called Kaggle, and Kaggle is a algorithm development competition platform. So basically the way we built this algorithm was we put 25,000 images into this space, divided between competition, like test images and reference images. And in this platform said, okay, folks, go, open to the public. We got 2,100 different solutions contributed, right? Or 2,100 teams contributed solutions and attempted this competition. We got to choose from the best one, and this algorithm was very, very specifically directed towards flukes. We are about to enter into another round of algorithm development, again with on the Kaggle platform. For this one, we have gathered 80,000 images of 27 different species to look at trying to develop an algorithm that can use any lateral view of any cetacean species, basically all the individually identifiable species features that any cetacean could present. So blue whales, right? They fluke, and their tails actually in fine resolution have quite a lot of individually distinct elements to them. But the reality is they don't fluke very often. So much better with a blue whale is the dorsal fin. So this algorithm, pieces of this algorithm, yes, you could retrain parts of it to do that. But I think what we're going to do is end up with a whole new set of algorithms to apply to different species. We do have a sperm whale fluke ID algorithm that's embedded into Happywhale. We don't have a lot of base data for sperm whales other than from the Azores. But I think the future is really bright here. And, we have the information architecture. If you imagine the algorithm, that's the engine, but you also need the hull, and you also need the linkage and the wheel and all the other pieces of the ship to get you from port to port, right? Rather than just the engine. So I'm pretty keen on the expansion of this. And to that end, big shout out to the Marine Mammal Center. We're just entering into a partnership with the Marine Mammal Center to implement this as it gets developed. So I'm excited about that.
Thanks, Ted. Okay, along the same lines, from Shari, how long have you been cataloging gray whales, and how many are currently listed in your database?
Shari, hello, and thank you for your, you made some comments on encounters recently. So with species other than humpbacks, most of the effort has been sort of, okay, so to basically put it in context, I have really myself, because of my PhD, been pushing like, hey folks, give us humpback whale flukes, right? And then what's happened is that in building this system, we've had folks say, oh, are you interested in other species in terms of the contributor side? And then we've also have researchers saying, hey, if you happen to get images of XYZ species, we are interested in those. And we've really tried to make the bridge between them, sometimes very successfully, and particularly in the Southern Ocean, where, I have strong ties into Antarctic tourism. People give us photos, right whales, blue whales, so on and so forth. We hand them on to the right whales, go to Jen Jackson, and the blue whales go to Paula Olsen. Well, the gray whales, because of a few contributors, I don't know off the top of my head. Maybe if I can multitask and search here real quickly, I could give you a number of gray whale ID'd individuals. But honestly, with gray whales, it's just been who has where people have told us IDs, we aim to put those IDs onto the individuals. For the most part, it's like, hey, if people have gray whale photos, we can gather them here. And then once we have the algorithm, once we have somebody championing a study using this system, then presumably we'll get a lot more. So I'm just looking, and there's 1,400 encounters in Happywhale and 100 identified individuals. So, considering the scale of the population, it's tiny, but it's kind of that, like, it could do, needs a champion.
Okay, and this is from Ellen. If a new image has no strong matches with a known animal, how do you decide whether the image is new?
Yeah, great question.
A new animal, or whether perhaps the image wasn't sufficient to definitely match it against a known animal?
Yeah, super great question. Well, I should have touched on that when I was showing you the little matching demo. Basically, that has been the time saver, right? It's great to find a match quickly, but what you really need to know is, hey, I didn't find a match. Is this animal new? And so what I took as the baseline for this is, hey, if we can do at least as well as Splash's manual matching, if we can find 90 or 91% of potential individuals, if we have an image, it's a good quality image, and we didn't find a match, and we have proven, with a known data set, that we're finding at least that high of a percentage of potential matches, then we'll just put a new ID on that whale that we didn't find a match to and move on, right? Because in continuing to search, we do continue to find duplicates. Now, I felt like that was a reasonable premise, because that's what we do in manually matching, right? We search and search and search until we're confident, hey, this is new. Those manually matched catalogs are hitting 90%, 91% of potential matches. Well, when we got to the place where, using a Glacier Bay National Park data set, this extremely well curated data set, thank you very much, Janet Nielsen, Glacier Bay National Park biologist, we found 97 to 99% of potential matches. So if the image is of a sufficient quality, and that's just you could show me an image and I'd tell you, oh, that's quality 2, not good enough. We would leave it unidentified. Oh, that's quality 3 or above, good enough. We'll put a new ID on it, and then forevermore be looking for that whale. Do we miss matches? Absolutely. But if we're only missing, 1, 2, 3% of matches, that's a very functional process, and we carry on with it. And that's what's allowed us to build up this 64,000 whale data set at this point.
Okay, another question. This actually doesn't refer to what you've been talking about. It's more directed to the local, actually the San Francisco Bay, and I can help you with this one. What local efforts are being done to prevent ship strikes in the SF Bay?
Izzy, or maybe Bill. Yeah, you guys.
Well,
Yeah.
Bill's not on, but I
Oh,
can
okay.
answer that. You know, we're part of the Marine Mammal Center, Bill, Mark, and I, and Tim, and there is a program or a group of people. Kathy George is the lead on this one, and they meet with Coast Guard, shippers, San Francisco pilots, and they're talking about what can be done to reduce/eliminate ship strikes in San Francisco Bay. And there's a lot of communication going on. Whenever we're out and we see a whale inside of San Francisco Bay, we notify the Coast Guard, and the Coast Guard sends information to the ships. It's just a general all alert bulletin. And we're looking at how to refine that, improve that. And the ships, you know, although whales are nothing in comparison to the weight of a ship, I mean, getting hit or hitting a 100-ton blue whale or a 60-ton humpback whale does do damage to ships. So there is something going on with a lot of different organizations on how to eliminate ship strikes in San Francisco Bay.
Yeah, and I might give a plug for our first speaker event of 2022, which is January 25th. And we will have a marine biologist from Point Blue. And that is what the talk is about, what we're doing about ship strikes.
Great stuff. I have so much. I mean, it's the greatest pleasure to be able to contribute something to that, you know, honestly. And Happywhale's impact on ship strikes, I think it's a bit indirect, but just to make the whales less out of sight, out of mind, I think is so important. And I have to say that in the course of this work, the scarring that I see on the whales that lived, right? And what we're not seeing is the scarring on the whales that died, other than like that one slide I showed with Michael Moore, the North Atlantic right whale. It's yeah, I mean, gosh, you know, people say, well, I thought these animals were smart. Why don't they get out of the way of the ships? I'm like, wow, do you dine on the middle of a freeway? I mean, why, you know, gosh, I mean, you might get hit if you had dinner on a freeway, right? And that's what we're doing, you know, so yeah, great work. Thank you guys.
Okay, a question from Era. You mentioned looking to create an algorithm using more of the whale's body. I have noticed with gray whales I send you that the ventral and dorsal flukes are distinguishable aside from the dorsal hump. Has it been noticed that the dorsals on humpbacks have some differences also?
So dorsal fin ID for humpback whales will be a very powerful thing. There are some examples of it. I have not seen accuracy assessments of it, and you know, and image recognition system is only as good as its accuracy, right? So but yeah, actually, that one study that I referred to using drone imagery, they are really keen to see dorsal matching, because more than 50%, just over 50% of the whales that they drone sample, they don't end up getting a fluke photo of. They do almost always manage to get a dorsal fin photo. Gray whales, you know, yeah, the flukes are great, but they often don't flukes. Humpbacks are great because a high percentage of them, especially on feeding grounds, will fluke, but mothers with calves on breeding grounds, good luck getting them to fluke. Really, really low fluking rate, right? So dorsals are distinct. We don't know over time how consistent that is. Male dorsals will definitely get heavily changed because, excuse me, in competitive behaviors that will bash against each other, you get the whole dorsal fin torn off. That's hard to identify from before and after it's torn off. But it'll be, you know, another tool in a quiver. I can't imagine it will be nearly as accurate as with flukes. But what really got me motivated to do this is that this algorithm is really powerful. It has weaknesses with partial flukes and highly rotated flukes, but very, very low image, very, very low resolution images. Literally, it's still matching them well beyond when I can confirm it, right? If I might have two photos, one of while we're approaching the whale, and the fluke photo is 100 pixels across, right? And then another full resolution fluke, and the algorithm will come up with a match with the very, very low resolution one, and I look at it, I'm like, I don't know. And then boom, there's the high resolution one that proves that it is the match, right? So the dorsal fins clearly have at least as much data in them as a 100 pixel wide fluke photo. So I'm very bullish about and believe that humpback dorsals, gray whale, the lateral view of gray whales, blue whales, fantastic. Fin whales are going to be more difficult because you have very little pattern. You only have shape. We have a set of beluga whales, and it's like, man, you want difficult image recognition. That's a real tough one. Nothing even to anchor it on. How do you know which end of the whale you're looking at? These they're tough, but we'll see.
Ted, one thing I could add about that is the humpback whales that we're seeing in San Francisco Bay, they don't fluke as much as animals that are out into the open waters. And so we've been trying to take photographs of dorsal fins and trying to match them when we do see flukes. So we have an animal identified by dorsal fin and flukes. And we're having some success, but we got a ways to go.
Yeah, and also dorsal surfaces of flukes. We've done a fair bit of, and I'm not saying that like hoping people will send. I mean, when people submit dorsal surfaces of flukes from, say, Monterey Bay, we pretty much ignore them because we get good ventral surfaces. But I can take a good vertical dorsal surface of a fluke, flip it, and then use the trailing edge to match, and it tends to work out pretty well. So
here's there was just a question from Jeff. Can you list the other species that you're trying to match with your algorithms?
Oh gosh, what are those species? I have a list here. I saw that pop up. Jeff Litton, what's up, my friend? Okay, I'm gonna have to do it off the top of my head because I don't have the list in front of me, but humpback dorsal fins, killer whales, there's a couple different ecotypes, but I'm expecting that the algorithm working on one will work on all ecotypes. We have some Cuvier's beaked whales, we've got belugas. I'm not at all hopeful that will work. Gray whales, fin whales, sei whale, Brutus whales, tiny data set there, same as sei whales. A number of species of Tursiops. We have spinner dolphins. There's I don't think we got spotted dolphins. The one set of individuals that I was sorry to not get a data set of was Risso's dolphins. But the idea here is to basically come up with enough diversity of species, you basically will accommodate all potential feature types, right? Shape, pattern, scarring, so on and so forth, such that we'll build this differently than how we've built with humpback whales, where we can basically say, okay, here's the training data set. This is, and it doesn't have to be a specific known species, same as one of those 27 species. It just has to have the same set of feature types, which should be any feature type that cetacean can present, because there's such diversity in there, and train it on that reference set of individuals and then match a novel set of individuals to it. So I'm hopeful that it's kind of the one algorithm to rule them all as far as a dorsal view of cetaceans. Success, I don't know, but boy, I sure didn't think the humpback fluke thing was going to work, and we knocked it out of the park pretty well. So should happen. We'll see.
So one last thing, Ted, and nobody really asked this question, but I know there are a lot of people who submit photos and of humpback whale flukes. maybe you can go over how people who have photos can submit them to Happywhale.
Yeah. Well, exactly. That's awesome. Totally jump past that. So yeah, I mean, the whole idea with Happywhale, actually, the origination of it was supposed to be like, oh, data from citizen scientists to scientists, and I didn't really expect it to become, quite frankly, the well-loved research collaboration tool that it has become, where actually the minority, a large minority, but the minority of data in here is from citizen scientists, but it's a very important data set too. It's quite simple, really. Just go to Happywhale, happywhale.com, and in the process, right at the top, it'll say submit images. You can create an account in the process of it. There are steps in there, and there's a couple links that show you what to do. If folks are like me, you probably aren't going to read the directions. You probably just put it in there. What's really important is date and location, right? And depending on the species, what kind of view is important, right? So with humpbacks, it's the ventral surface, the underside of the fluke. With gray whales, it's that lateral view, right? With blue whales, it's dorsal fin and lateral view, and so on and so forth. But there's some material there. If you click around a bit, you can get that. But yeah, it's just basically, we want to know the date. We want to know the location. We prefer to know who you were out with. That gives some context. If you say, oh, I was in Hawaii with Monterey Bay Whale Watch, we're like, yeah, you actually don't have a clue where you were, do you? But if you say, oh, I was in Hawaii with Steve's rafting, like, okay, cool, I know that. Okay, we got that. It gives a little context. And the best thing of all is, make sure your settings, if you're going whale watching, make sure your camera is set to the right date and time. If you have GPS, please use it. GPS enabled cameras, like if I could be Santa Claus, every little chimney of every whale watcher in the world would get a GPS enabled camera just dropping right down it. So yeah, that stuff because we auto read that on the import, and it actually makes it a lot easier on your end too.
So speaking of that, do you want the original photo? Do you want a crop photo? What is best for you?
Cropped is great. If JPEGs, not raw. We don't do raw. Just image size. There's just no reason to have that massive file, and there's so many different file types and so on. But JPEGs. I guess a big thing for me is don't let perfection be the enemy of the good, and that is, someone who's got, I've got thousands of photos, but oh gosh, I need to crop them all. Oh, I'll get to that, and they never do. Right. So if, grab the flukes and, send us a few, and we'll see what we find. And hopefully, our goal has been make it easy and make it rewarding. So if it is easy enough, you'll come and try it. If it's rewarding enough, you'll come back and you'll say, well, that was really cool. Okay, I'm going to dig deeper. And quite frankly, because of everyone locked up at home over the last year and a half, we've seen a lot of that, the sort of the 10th tier project. Oh yeah, the stuff that's on the hard drive. So finally, folks cooped up and not able to go whale watching went and dug up all those images, and it's really, honestly, it's really benefited our data set. So it doesn't have to be cropped, but yeah, sure, that's great. We do a lot of cropping here.
Okay.
Yeah.
Well, I think I've gone through all the questions, including the questions I had. Susan, I'll turn it over to you. Maybe you have a question or two you'd like to ask.
No, I think we covered a lot of ground. It's just so fascinating. I guess the only thing that would be of interest to me, you're really kind of a mix of an entrepreneur as well as a marine scientist. So, maybe just, what you started TED in 2015, and what were some of were there hard times in the journey? I mean, what were some of the challenges? Maybe just real quickly would be of interest.
Well, I started this for a challenge, right? So, challenges are kind of the attraction. I mean, I've been guiding. I went to grad school. It's conservation science. Yay, yay, yay. Oh, got a young family, got to feed them, and a really awesome job opportunity working with my family's wildlife safari company. But it was always this, okay, I'm going to get back to science. And I kind of had a few meanderings, but this has really been the dive back into science that's allowed me, it's actually really, really rekindled my love of guiding and sharing the knowledge of these wonderful places that I've had the pleasure of guiding to having a little bit more and very personal insight. But I mean, honestly, the biggest single challenge, not unusual, has been money. Well, quite frankly, I was in a good place initially. I self-funded this initially. Then we got a really good grant from booking.com of all kind of random sources. But I always kind of had this like, okay, we're not going to pull from the traditional, conservation science sources. And I had this really great expedition cruise tourism sponsorship that was like a 5-year deal, all ready to, pens already, and then COVID hit, and they're just like, sorry, guy, and so yeah, it's been a big struggle. We're doing all right for now, but what I'm really trying to find is, trying to find sort of lasting long-term stability, because right now, I mean, honestly, one of the scariest things to me is we have built this with this notion of archival data that in 50 years you could say, hey, what were the whales like back when the waters were so cool as they were in 2020, 21, right? But if Ken, my programmer partner, if either of us were proverbially or really hit by a bus, this whole thing would be in an existential crisis. And so looking forward, to me, that's the biggest challenge, is to push this thing into kind of stability and sustainability. It's basically mostly money that does that. And I just saw, maybe, I don't even know if that was a chat or an email, but I saw Bonnie just posted or just sent me Michael Moore's book title, We Are All Whalers, which is something to look up. I definitely would encourage it. Yes, it's depressing, but it's also hopeful.
Another really important question to you.
Mm-hmm.
How can people financially support Happywhale?
Oh, bless you. Well, okay. So,
that wasn't my question. Somebody asked that.
Well, okay. Two things I want to say is, like, look, Happywhale, I mean, I love support, but it's also, we're all a community, and American Cetacean Society is super legit. And, so if you have the motivation to support someone, I'm just as happy as you go to American Cetacean Society. But Happywhale is, we're closely partnered with a group called Whales of Guerrero. It's a community conservation science group that's a US 501c3. I can type it, Whales of Guerrero, Guerrero, there in the chat. Oops. Yeah. Did I send that to everyone? I think I just sent that to you.
Just to the panelists, yes.
Okay. Let me try that again. So Whales of Guerrero acts as a fiscal sponsor to us. So if you want to support us, and you want it to be tax deductible, just Google that and make a contribution there, and we'll love you. If you want to name and adopt a whale, our whole thing is, if you want to name and adopt a whale, make a contribution to conservation science. It doesn't have to be us. We ask for it, but I feel that that's like a minimum donation. We're asking for a minimum donation of $500. It can be to ACS, it could be to Whales of Guerrero, it could be to any conservation science entity of your choice. And with that, we can name and adopt a whale. I think it's super cool. I saw someone ask, who's the most famous whale? Well, what matters to me is who's the whale that means the most to you. It's like we go out and we see a whale and we have this amazing, drawn to tears kind of beautiful moment. And then that whale swims off and it's, we might be able to make that whale not anonymous, not just a number, right? I actually got a manuscript of a book, and from many of folks here will have seen The Whale Detective, and Tom Mustill, the director of that book, director of that film, has a book that is coming out, and in the dedication he says, and I don't remember the number, but he says, for CRC 12537, who started me on this journey, or something like that. And I'm like, really? You could use a nickname, but it means a lot to have a nickname with a whale. Yeah. So.
Well, it's just been such a fun evening, and so appreciative of your mission and all of your efforts, and love to have you come back maybe next year at this time, Ted. And yeah, and with that, again, thank you to our community, and we'll see you in the new year. We start off on January 25th, so 4th Tuesday of the month. Thanks, everybody.
Thank you all.
Good. I stopped the recording. So,
okay.
Yeah, anyway, wow. So fantastic. A lot to take in.
Well, it's good fun. Thanks for, it's great. Thanks for hosting it. You guys are fantastic. I love ACS, and yeah, fun to do.
Marine Mammal Center also sponsors naming whales. And we're also looking at, we've identified 120 different dolphins in this area, and we have about maybe 15 animals that haven't been named. So we're thinking of doing that to increase funds.
Cool.
But
I think, yeah.
If you ever get up here, we have a boat that we use to do surveys in the bay. Maybe in the spring, if you have a day you come up, we'll take you out in the boat in the bay, and maybe we'll be lucky enough to see some humpbacks.
It'd be fun to do. I would love that.
Yeah.
I will absolutely take you up on that.
Oh,
for
please
sure.
do. Please do.
Cool.
Okay. Good night, everyone. I'm going to end it now.
Bye-bye.
Okay.
Bye.
Take care, all.